Hi class, in this recording we are going to be focusing on platelets, hemostasis, and coagulation. As we look at this process of hemostasis, hemo means blood, stasis means to stop or maintain. So if we look at this, really the root words of this mean exactly what it's defined as. The cessation of bleeding. Make it so that we don't leak. We don't want to have a fatal loss of blood. And as we're bleeding, and there's not a hard and fast definition I could find in terms of the volume of blood you need to lose, but say air quotes here, if you have excessive bleeding, you are hemorrhaging. And as we're looking at this process of hemostasis, there's three mechanisms that we're going to use. We have the vascular spasm, platelet plug, and blood clotting, aka coagulation. And as we look at these three mechanisms in more detail on subsequent slides, it's worth emphasizing that platelets, aka thrombocytes. So a platelet is also known as a thrombocyte. Thrombocytes are going to play a role in all three of those mechanisms. So here we have the platelet, also known as the thrombocyte. And a thrombocyte is just going to be a small fragment of a different cell type called a megakaryocyte. So here we can see a photomicrograph of megakaryocyte, and this is kind of difficult to distinguish because of all the false color given to it. But as we look at this megakaryocyte, I think it's a little bit easier to look at the figure at the bottom of our screen. Here's our red blood cell for comparison. And here's a white blood cell. This is a neutrophil in particular as we look at that white blood cell. And these cell types, if our red blood cells are kind of our measuring stick when we look at blood tissue, photomicrographs, the megakaryocyte is ginormous relative to those cell types. And it's constantly exuding pseudopods into the bloodstream, blood capillaries. And as it exudes those pseudopods into the blood capillaries, the pseudopods will fragment. And as they fragment, they turn into individual platelets or thrombocytes. As we look at the platelet, it's going to have a very interesting, very complex internal structure that's going to have lots of caniculi or canicular system within it. So it's going to have lots of small passageways within it. And as we look at the platelets, they are capable of a little amoeboid movement, not that much. Typically, platelets are just going to go with the flow. They're kind of like the inner tube flowing down the Chippewa River in Eau Claire. And as we're looking at a normal platelet count in a human being, it's as low as 130,000 to as high as 400,000 platelets per microliter of blood, just to give you some baseline numbers as a point of reference. So what do platelets do for us? Well, they help us not to bleed a lot, really to just summarize everything. So they're going to secrete chemicals that are vasoconstrictors. Vasal referring to the blood vessels, constrictors referring to the diameter of the blood vessels getting smaller. So if you have a leaky pipe and you make the pipe smaller, the pipe won't leak as much liquid out. I recently redid all the plumbing in my house, so I have lots of plumbing analogies I could share with you, particularly when we get to the blood vessel unit. As we look at platelets, they stick together. And we're not talking about how they back each other up emotionally. I mean, they literally, physically stick together. They form a platelet plug. So in addition to making the tube smaller, the blood vessel gets smaller through vasoconstriction, they're also going to block up the hole itself. And they're also going to secrete procoagulants, chemicals that promote coagulation. And they are also going to initiate the formation of a clot-dissolving enzyme, which seems kind of counterintuitive. Why would a platelet promote clotting, and then also initiate the process of dissolving a clot? And if they were to do that at the exact same time, that would not make a lot of sense. Some of these chemical reactions are time-dependent chemical reactions. And that's how a platelet can both promote clotting and promote the dissolving of a clot. When we look at platelets, they're able to attract neutrophils and monocytes to the site of inflammation because, generally speaking, if you get a cut, that cut had to come from the outside of your body to get to the inside, and lots of bacteria have been introduced. So we need the neutrophils to get rid of those bacteria, and there may be bits of foreign debris, and the monocytes will transform into a macrophage and help get rid of that foreign debris. As we look at platelets, they help our white blood cells to phagocytize and destroy bacteria, particularly the neutrophil and especially that monocyte. It has assistance from the platelets for phagocytosis, and then also our platelets are going to secrete growth factors so that we can upregulate tissue regeneration at the site of injury where our blood vessels are damaged. Now, as we look at the process of making platelets, that's called thrombopoiesis. Thrombo for platelet, poiesis for production of. So we talked about erythropoiesis, leukopoiesis. Now we're at thrombopoiesis. So we have stem cells, multipotent stem cells, adult stem cells that are going to receive thrombopoietin. And from there, when those stem cells are stimulated by thrombopoietin or poietin, they differentiate into a megakaryoblast. The megakaryoblast is then going to just replicate its DNA over and over and over and over and over again. So it has an enormous nucleus. And those gigantic cells are called megakaryocytes, in part because they're so large. And they have enormous multi-lobe nuclei that are about 100 microns in diameter. So if you had really good eyesight, you might be able to see a megakaryocyte nuclei with your naked eye. I remember when I was an undergraduate, I had better eyesight than I do now. I still have pretty decent eyesight. I don't need to wear my cheaters yet. But I remember as an undergraduate, I could look at 200 micron objects and just barely see them with my naked eye. So that 100 micron object, maybe you could see it if you had awesome eyesight. Well, Aaron saying yes. Well, as we look at these megakaryocytes, the megakaryoblast that make the megakaryocytes will then have the megakaryocytes differentiate and produce those platelets that we talked about, those thrombocytes. There are going to be blood sinusoids, or little hollow cavities next to the blood vessels that the megakaryocytes are going to reside within, and the megakaryocyte will have pseudopods extend into the bloodstream and then form protoplatelets, and then those protoplatelets will start to fragment and turn into matured platelets. And those platelets typically last for 5 to 6 days, and most of our platelets are going to be stored in our spleen. We have a reserve of platelets. So I shouldn't say most, I would just say a good fraction, a high proportion of our platelets are stored in our spleen, and they act in reserve for us. So let's say we have a cut. We need to stop bleeding. We need to cease bleeding. Step number 1 is vascular spasm. Oh, let's go back a slide there. I accidentally advanced. Step number 1 is our vascular spasm. So we're going to have vasoconstriction of the blood vessel, and that is going to be the single most immediate way that we can stop losing blood, make the blood vessel smaller. What causes the vascular spasm? We have some pain receptors that can directly innervate or directly send electrical signals to the smooth muscle of the blood vessels. If we have any injury to the smooth muscle tissue reacts by constricting, and the platelets themselves can secrete serotonin. That in the brain, serotonin is a common neurotransmitter. In this context, in blood vessels, serotonin acts as a vasoconstrictor. So what happens when we have that happen? We have the blood vessel get smaller in diameter. The pain receptors can cause vasoconstriction for a few minutes. Smooth muscle injury can cause, tens of minutes to up to an hour. And that provides us just enough time for the other 2 clotting pathways to get their act together and plug the hole. We also are going to have the platelet plug form. As our platelet plug forms, that is going to be a large mass of platelets that clogs up the hole in the blood vessels. Our intact blood vessels have a very smooth layer of cells called the endothelium on the inside, coated with prostacyclin that repels platelets. But what I really want to emphasize is that when we damage the inner lining of our blood vessels, all of a sudden we have collagen exposed to the blood plasma. And when that collagen is exposed to the blood plasma, the platelets will stick to the collagen, and the pseudopods of the platelets constrict drawing together other platelets and knitting together and pulling together the injury to make a platelet plug. These platelets eventually will degranulate. When we say degranulate, they release all the chemicals they were storing inside. Some of the things they release include serotonin for that vasoconstriction aspect. They are also going to release ADP, adenosine diphosphate, and that's going to attract even more platelets to get stuck to that platelet plug, and they'll release thromboxane A2, which is an eicosinozoid. This is a chemical that specifically is going to promote platelet aggregation, degranulation, and even more vasoconstriction. So this is a chemical that helps other platelets to recruit other platelets even better. So platelet sticking to each other causes even more platelets to stick to each other, which causes even more platelets to stick to each other. It's a positive feedback cycle, and we need something to break it. And to break this positive feedback cycle, we need to seal the tear in our blood vessel. So that's coagulation or clotting. So the platelet plug is not the same thing as clotting. The platelet plugs is just a big glob of platelets plugging a hole. Coagulation is going to involve a fibrin mesh. Give me a second here. We'll talk about the shoes in a moment. So we need the fibrin mesh, and you can see that fibrin mesh kind of crisscrossing the platelet plug over here. So once we take fibrinogen, which is the deactivated form of fibrin, we activate fibrinogen, turn it into fibrin, and all of a sudden, miraculously, all of a sudden, we go from no fibrous proteins to all of a sudden these fibrous proteins precipitate out of the blood plasma around the platelets. And the platelets themselves can release procoagulants, and some of these procoagulants will already be waiting in the bloodstream. They've been released by the liver into the blood plasma, and one chemical will activate another chemical, which will activate another chemical, which will activate another chemical. It's a reaction cascade, and we'll talk about them in just a couple slides. These two reaction cascades are the extrinsic pathway and the intrinsic pathway, and they get the name extrinsic and intrinsic pathways based on how they are initiated. The extrinsic pathway needs to have a foreign object from outside, from the exterior, enter into the bloodstream. So you need damaged tissues for this to occur. You need something from outside going to the inside to physically damage the tissues. Our extrinsic pathway is a 2-protein cascade. And then we have the intrinsic pathway. You can initiate this process of coagulation without physically damaging the tissues of your blood vessels. And that's because of a pathway that's initiated from the inside, the intrinsic pathway. And this pathway involves 4 proteins working together. So as we're looking at these two pathways, the extrinsic and the intrinsic, I like to think of them like these shoes. We have the extrinsic pathway over here, a nice pair of navy blue flats. It's nice and simple. When you're wearing a pair of shoes like this, you can be pretty confident that not much will go wrong. Not many things go wrong with your shoes when you're wearing a pair of shoes like this. And this is kind of like the extrinsic pathway. It's simple, it's short, it's sweet, it's to the point, it's very functional. And then we have the peacock stiletto over here. And when we look at this peacock stiletto, this reminds me of the intrinsic pathway. And as I look at this peacock stiletto, I think to myself, oh my goodness, there are so many things that could go wrong when you're wearing a shoe like this. I mean, you could snap the heel right there, the feathers could come off, they could come untied. I mean, you could just monologue for a long time about how shoes like this could fall apart in many different ways. And that's kind of like the intrinsic clotting cascade. The intrinsic clotting cascade is more complicated and is more prone to error because there are more steps. So let's look at them in more detail. We have our extrinsic pathway right here. We have some kind of foreign object damaging the tissues. And that's going to cause factor 3 and factor 7 to interact with each other. And then calcium ions in the bloodstream are then going to then cause those two factors to activate factor 5. As we look at factor 5 is the first clotting cascade factor of the, I'm going to put a C, actually I'll write it out here, of the common pathway. Both the extrinsic and the intrinsic pathways feed into the common pathway. And as we're looking at these pathways, let me clear the screen here. The intrinsic mechanism is going to involve 4 proteins. We'll go from factor 12 to 11 to 9 down to 8. And yeah, as we're looking at this, it's that nice. It goes in sequential order. It's a countdown. There are other names for these factors. One of them is called the Christmas tree factor. That one always stuck with me from when I was an undergrad. But I'm a much bigger fan of the Roman numeral system, the factor 4, factor 5, factor 3, factor 7, because it's easier to remember the steps in order. So what do I want you to know for the test? I want you to know extrinsic is simpler. It involves 2 proteins in the cascade. Intrinsic on this side. Intrinsic on this side is more complicated, and it is going to it's going to not involve damage to the tissue. But either clotting cascade pathway will ultimately lead to factor 5. And this, just as a personal reference here, my father and several of my siblings carry a factor 5 mutation that causes their factor 5 protein complex to be activated abnormally easily. So for my dad and several of my siblings and several of my aunts and uncles, they have had pulmonary embolisms. They have lots of blood clotting issues. So they are those individuals that when they have to fly in an airplane, they typically need to fly first class just so they can have enough leg room so they can minimize the amount of deep vein thrombosi that occur while they're flying in that airplane. As we look at factor 5 is going to involve many different things interacting with each other. That factor 3, factor 5, calcium ions, and phosphorus trifluoride are all going to, PF3, phosphorus trifluoride, are all going to interact with each other to form and move to the prothrombin activator. And I've been saying factor 5 here. I need to apologize. I apparently can't read Roman numerals. I mean factor 10, X for 10, V for 5. So factor 10 over here. Factor 10, I'll write it out with Arabic numerals. There, factor 10. We'll then move down here to the prothrombin activator. And that prothrombin activator interacts with factor 5. Factor 5 causes the prothrombin activator to activate factor 2. And factor 2 will then be converted to thrombin. And instead of saying factor 2, I like sticking with prothrombin. We go from prothrombin to thrombin. Prothrombin is the deactivated version of thrombin. And then thrombin causes factor 5 to give us more prothrombin activator, which causes there to be more thrombin. So right here, this segment right here is a positive feedback loop. It's a cyclic cycle that is self-amplifying. And then thrombin is going to take the deactivated version of fibrinogen. Thrombin turns fibrinogen into fibrin. And then fibrins will interact with factor 13 to form a polymer that will then knit all of those platelets together. And then next we're going to have coagulation. As we look at this last step, coagulation is going to involve the fibrin mesh. So I've spent a lot of time talking about it on the previous slides, so we're going to breeze through these next 2 to 3 slides. With that extrinsic pathway, it's going to be initiated by thromboplastin, factor 3, from the damaged tissues. From factor 3, we go to factor 7, then 5, and then to 10. So extrinsic has fewer steps. The intrinsic pathway is going to be initiated when platelets release factor 12, also known as Hegeman factor, or Hageman factor. And from factor 12, we go to 11, then from 11 to 9, 8, and then to 10. And then as we look at all these cascades, they all require calcium ions to be present. And this is one of the reasons why we need to maintain blood homeostasis for calcium. If we don't have enough calcium in our blood, not only do we have trouble sending action potentials in our nervous system or constricting muscles, we also have a hard time clotting our blood. So here is, oops, double clicked there. Here's our cascade in order. We go from factor 12 to 11 to 9 to 8, back up to 10, then from prothrombin activator to the thrombin, and from thrombin to fibrin, and then the fibrin does the coagulating itself. In terms of coagulation, factor 10 is where we have the intrinsic and extrinsic pathways converge. And this is going to lead to the production of that prothrombin activator, which converts prothrombin into thrombin, and that thrombin will take fibrinogen and convert it into fibrin monomers. And those monomers we need to link up with each other. It's like snapping a bunch of Lego bricks together. And we go from fibrin monomers to a fibrin polymer. And as we're looking at those fibrin polymers, factor 13 in particular will take those fibrin polymers and cause them to connect to each other. It cross-links the fibrin polymers. This coagulation process has a lot of positive feedback. Thrombin makes it so that we have more prothrombin activator, and I talked about that 2 or 3 slides ago. And as we look at this process, we can quantify the efficiency of coagulation by making a 1 millimeter deep incision in a patient and then timing how long it takes for that person to stop bleeding after we make that 1 millimeter deep incision. That's all we have for this recording on the platelets and coagulation and clotting cascades. If you have any questions or comments, please feel free to post them in the class discussion boards or to shoot me an email. And as always, happy studies.